Hello again, Zoology 2700 enthusiasts, and welcome to the rest of my introductory thoughts about platyhelminth worms. And I'm doing this in a kind of post-lecture format so that you can graze it at your own pace in an asynchronous way, but my suggestion is you try and watch a little bit of it at least before your labs this week, because we try and keep the labs and the lectures kind of roughly in sync. And so here we go. We're going to talk about the platyhelminth worms. And remember, this is by far, if you take a look at that, the diversity of the platyhelminth worms within the Spiralia, within the Bilateria, this is by far and away the most diverse group of bilaterian that we've dealt with so far. It's not as diverse as the cnidarians, as you can see, but it is diverse. And they exhibit a whole bunch of different strategies associated with living in a marine environment, in a freshwater environment, and in a terrestrial environment, as well as living parasitically. Now, this is one phylogeny, a simple phylogeny that you could imagine me presenting to you as to stimulate a quiz question. It's slightly different than the figure that Brusca et al. Use, but consider, for example, platyhelminth worms being the large kind of green box on the outside. Now, Turbellaria, or planarians, they're put in those Tribbiani quotes because it's not a monophyletic group. I've actually made mention of this in lecture before when talking about Xenocoelomorpha and the acoelomorph part of the phylum that's made by combining Xenoturbella and the acoelomorpha. We used to think that the acoelomorphs were planarians. So Turbella is essentially here used to represent the free living. Now, within the Neodermata, this is the parasitic part, there are monogenean cestodes and trematodes. Maybe you haven't been introduced to the monogenean neodermatid platyhelminth parasites, but I bet you've heard about, even if indirectly, tapeworms and liver flukes. And those are two examples. Now, free living platyhelminthes look a little bit like this. This is a video from Sherry's Twitter account last year, and maybe 2 years ago, but it's a little bit of a stream of looking at a live planarian that would have been in the lab. Now, apomorphies for this group, for the platyhelminthes, include multiciliated epidermis and gastrodermis, the protonephridia, the excretory organ that we talked about today, and that they're hermaphrodites. There's about 20,000 species of them worldwide, and about 70% of them are parasitic. And you'll see our emphasis on the group is biased towards those parasitic forms because that's where the majority of the diversity lies. So we talked today in lecture about the protonephridia, that there are these ciliated cells that beat here. This is the figure you've seen already in lecture, and this is the kind of a corresponding figure in your text, where there's a negative pressure created by a beating flame cell that's characterized by one or two cilia, and that this beating creates negative pressure, draws out any byproducts of metabolism, and allows the excretion to the exterior environment. Now, to give you one of the things that I wanted to do today in this asynchronous portion was to give you a link to an online tool that you can use to take a look at this in action and show you a video of what they look like in real life. So in today's chat, I put this link to the University of Alberta. And here's, you can play with some of the parameters online, but here's a look at basically what we're seeing. And I've made a video of theirs, and so you're looking at the protonephridium here. I'm pointing, that's very helpful. I'll use and you've got the fluid flow created by the negative pressure that's into the protonephridia, and then different amino acids, wastes, spent hormones, and water follows that gradient of pressure into and then out of the organism. Now, why is it called a flame cell? Well, here's what it looks like if you were to kind of take a section of that planaria and look at it under the microscope. And you can see, if you kind of squint your eyes, or if you're me, if you remove your glasses, that basically that flickering of the cilia in that protonephridia cell kind of looks like the beating of a candle flame, so a flame cell. Now, the musculature that we can see in the Platyhelminthes is our first example, first kind of large example of a successful bilaterian lineage, is much more complicated than we've seen to date. So they have circular and longitudinal muscles. We've seen that dating back to the Cnidarian. But they also have complex dorsal-ventral muscles, which permits this amazing kind of fluid swimming action. So this is a free-living marine flatworm, platyhelminth flatworm, that you're seeing there moving through the water. And it's allowed that complicated musculature. This is no longer just a hydrostatic skeleton, as you would have by the antagonistic longitudinal and circular muscles squeezing against each other, but you've got these dorsal ventral muscles extending across dorsal ventrally, as you might imagine, across the body that permitted this undulation, a dorsal ventral undulation to happen. Now, free-living planaria, you may have run into in your own training as a biologist to date if you've worked with flatworms and cut them into pieces. Now, this was actually done by someone named Thomas Morgan, who won the Nobel Prize for Physiology or Medicine in 1933. In and amongst other things that he did, he showed that you could cut the flatworm into a maximum of 279 pieces, and each one would grow into a new planaria. His Nobel Prize was not exactly for the planaria experiment, and one must imagine that there was a graduate student who worked at cutting up 280 slices, and perhaps that experiment failed, because 279 is the word in the literature. Just like their musculature is complicated, so is their nervous system. So we see nerve cords and nerve nets, and as we talked about in general with the bilateria, we see the concentration of these nerve cords into cerebral ganglia. So perhaps not a true brain as a pinky in the brain, but it's at least pinky in the cerebral ganglia. They have lots of complicated ways of elegant ways of responding to their environment. They have statocysts embedded in their wall that are gravity receptors. They have ciliary receptors that are mechanoreceptors for bumping into things, and different like rheoreceptors shown there, or chemoreceptors as well to respond to the chemical environment. They have photosensitive cells. They have pigmented cells. This is a free-living marine planaria again, and that little line with the white text, that's showing you essentially a pigmented eye spot, which is a concentration at the anterior end of these photoreceptors, and it lets the planaria assume strongly negative phototactic responses, that they move away from light. They have a mouth, but no anus. They have a sucking pharynx, and that's what you're looking at here in this video, is the mouth of the planaria kind of reaching out, and the sucking pharynx kind of moving on top of little bits of chunks of liver or things that it might be eating inside of that aquarium. Lateral branches of the gut and phagocytic gland cells, so implying two things. The first is that complication and diversification inside of an increasingly large body as a metazoan. We've talked about the pros and cons of being a metazoan and now a bilaterian. These are two of them, so specialization. And then lateral branches here, as you can see in the gut, implying a response to a need for an increase of surface area. So increased lateral branches, increased microvilli, these are all things that permit is evolution's way of solving a surface area to volume problem. Now, some of the mating system of free-living planaria you might have seen before. This is a National Geographic Wild about the world's weirdest flatworm, where they engage in showing you 2 minutes of the world's of flatworm penis fencing. So remember they're hermaphrodites, so insemination essentially involves a penis and two flatworms fencing, and the penis piercing the dermis, just the body wall, and then inseminating. And so in those two dancing or mating hermaphrodites, whichever one is successful first, that sperm inseminates the other, and so the pierced one assumes and begins to grow the offspring. Terrestrial flatworms can be predatory and can be up to 2 feet long. This is one, I believe, from French Guiana. But they're moving around the world, as many invasive species do associated with agricultural industry and potted plants. This is the hammerhead flatworm, and a Twitter post and associated with by a Peer J publication associated with the first record of this Bipallium adventindium arriving in Montreal in May of 2018. Now, the last kind of little bits that I'd like to talk to you today in this pre-section are about one of the key adaptations of the parasitic flatworm that it has, parasitic flatworm species that they have to being parasitic and to living inside of their host organism. Now, for the Neodermata, one of the key aspects, key adaptations towards living parasitically is this neodermis. So it's a ortegumen, might be the other word that you'd see it referred to as. So this is the body wall. You're looking at figure 10.7 here. Chapter 10 is the one to refer to for the Platyhelminthes. And in the body wall, you're seeing the cross-section of the neocytium here, the tegument, which has a syncytial zone. Now, the syncytium is an adaptation that gives an organism, it's essentially a sharing of cells, of cell wall contents that allows rapid sharing, absorption, and sharing of different nutrients across that body wall. You'll see also some other adaptations towards living parasitically and responding to a surface area to volume problem, in that they have extended the surface area of the outer wall with these tiny microtikes. And so these increase the surface area. And so if you imagine this being embedded, if this is a tapeworm embedded in your gut, or a liver or a fluke kind of moving around your body or embedded in your liver, this is increasing the surface area that the parasite has to gain nutrients from your blood or your tissue. And then the syncytial zone is an adaptation towards sharing that amongst the organism and amongst the parts of the organism. Unlike the free-living Platyhelminthes, the parasitic ones are not ciliated. They're not about moving. And so the syncytium gives that the organism tight control over what enters and exits its body. So this syncytium is a multinucleated mass of cytoplasm that's not separated, as I said, into individual cells. And so this creates a distal anucleate cellular layer that combines all these elements and allows the rapid sharing across the length of the body. And that's extended by these cytons that connect into cytoplasmic extensions that move the length of the body. And there's the main body of a tegumental cell, the neodermis tegumen. And that is about all that I wanted to say, but I wanted, before you met in lab, to talk about these adaptations and apomorphies that characterize the Platyhelminthes. We'll see you next time.